Have you ever cut your hand before and wondered how your body heals that wound? Your body has this amazing ability due to the cell cycle. The cell cycle is a series of events that lead to a cell to create two daughter cells, effectively duplicating itself. The cell cycle can be broken down into three main phases: interphase, mitosis, and cytokinesis. Let's first break down interphase. There are also three main phases of interphase: G1, which means gap 1, S, which stands for synthesis, and G2, which means gap 2. During G1, the cell synthesizes many enzymes and nutrients needed for cell division, such as phospholipids, which are needed to create a second cell membrane later on in the cell cycle. Also, the cell begins to grow in size during G1. During the synthesis phase, replication bubbles start to form in the chromosomes. These bubbles merge with others to form two sister chromatids, which are the templates for new DNA strands. Nucleotides with the complementary nitrogen base are added to the DNA strand, adenine with thymine and guanine with cytosine. These chromatids are made up of chromatin, which is a histone DNA complex. These histone proteins compact and package the chromatin into chromosomes. During G2, the cell grows a little more, has a chance to make more proteins, and it duplicates its organelles like chloroplast and mitochondria. The next phase of the cell cycle is mitosis, which is broken up into four main parts, the first being prophase. During prophase, the chromosomes condense. The centrioles move to each side of the cell and form spindle fibers. Also, the nuclear envelope begins to disintegrate. Second is metaphase, where the nuclear envelope completely goes away. The spindle fibers align the chromosomes lined up in the center of the cell by connecting to the centromere. Third is anaphase, which is where the centrioles pull apart their chromatids using spindle fibers at their centromeres, putting half of the chromatids on each side of the cell and beginning to split the cell in half. Finally, we have telophase, where a nuclear envelope begins to form on each side of the cell and the chromosomes uncoil, and an indentation of the cell's surface called the cleavage furrow forms. It should be noted that plant cells do not form furrows because of the rigid structure of the cell wall. Cytokinesis is the third part of the cell cycle. This is where the cell finally splits into two daughter cells. The nuclear envelopes are formed on either side, and a ring of protein filaments called a contractile ring forms which pinches the plasma membrane until the cell splits in half. A plant cell does not split using a contractile ring because the cell wall cannot be pinched. Instead, vesicles form in the center of the cell, which eventually grow into a cell plate, dividing the cell in half. You might be asking, what would happen if a cell divided uncontrollably, without restraint? Well, this is called cancer. Cancerous cells may divide way too fast because they bypass checkpoints along the cell cycle. They also do not communicate with other cells, and they do not carry out normal cell functions. Your cells may become more at risk to become cancerous if you are exposed to radiation or if your genetics make you more susceptible to have cancerous cells. Cancer is deadly because it hinders your body's ability to perform certain functions by forming tumors.